In this video, we want to talk about how stuff crosses the cell membrane. Keep in mind that when you're looking at a cell, you've got a situation where there are times when you want to get things from the outside of the cell to the inside of the cell, and other situations where you want to move things from the inside of the cell to the outside of the cell. So the question then becomes, well, which mechanisms do we use to get that done? And so that's going to be our learning objectives for this video. So number one, we want to describe the difference between simple diffusion and something called facilitated diffusion. Now, simple diffusion, you actually already know that. In the past, I only called it diffusion, but now I'm going to describe it as simple diffusion. Then we're going to talk about these two things called primary active transport and secondary active transport. And finally, we want to discuss this thing about called cotransport and the difference between symporters and antiporters. Now, before we get started, you need to be able to refresh your memory and understand what a concentration gradient is. If you don't know what a concentration gradient is, please go back to previous lectures and just make sure you're very comfortable with the idea of a concentration gradient. Remember, gradient just means difference. So it's a difference in concentrations, which by definition is the parts of something per unit of volume. And so make sure you're comfortable with that. Otherwise, these things aren't not going to make any sense. So here's what we want to do. We want to talk about, again, diffusion, primary active transport, secondary active transport. So first off, diffusion. Well, you already know diffusion is the movement of stuff from high to low concentration. And when that happens, it's moving down its gradient. It's moving down its gradient. Now, there are two types of diffusion that we care about, simple and facilitated. Now, the one that you already know, that you've seen in images like this, where stuff moves from the left here to the right, according to its concentration gradient, and moves from high concentration, low concentration. These pictures are actually indicating, and again, this should look familiar to you as well, this is indicating simple diffusion, meaning that there's no protein here that's required. If that doesn't quite make sense, let's go ahead and actually talk about facilitated diffusion, because what facilitated diffusion is, it's diffusion that occurs with the help of a protein carrier or channel. Now, when I say help, keep in mind, it just means that the protein is there to move things along. It doesn't mean that it's pushing things against its gradient. In both cases, remember that diffusion by definition is the movement of stuff from high concentration to low concentration. So let's look at this. What we have here on the left is simple diffusion. Just to be clear, this is the outside of the cell, this is the inside of the cell. And this book is telling you that you got a high concentration of stuff on the outside, low concentration of stuff on the inside. Now with that, when stuff just moves from high to low concentration, crossing that barrier, whether or not you cross a barrier, it doesn't really matter. But in this case, crossing that barrier, the cell membrane, this is called simple diffusion. So what is facilitated diffusion? Well, what we see here are a protein here and a protein right over here. Now this one happens to be a channel, this one happens to be a carrier, the differences of which we don't particularly care about right now. But I want you to see in both cases, these are called facilitated diffusion. Now, this one is channel-mediated facilitated diffusion, and this one is carrier-mediated facilitated diffusion. You know, for what we do in our course, I'm not going to make a big stink about whether it's channel-mediated or carrier-mediated, because for our purposes, they're essentially the same thing. At the end of the day, you got a protein here that's going to help you move things from outside to inside. To say it differently, in this case, high concentration to low concentration, which in this picture is outside to inside. So these are facilitated diffusion. So pretty simple and straightforward. So when you see something like this, again, this is simple diffusion. This is simple diffusion. This is simple diffusion. Now, if I were to put a protein here and tell you that, now these purple dots cannot go from chamber A to chamber B unless they pass through this protein, then we now would call that facilitated diffusion. But if you don't actually need that protein there, it's just straight up simple diffusion. Okay, so not a lot of new information here. Again, you already knew about simple diffusion, but now you know about facilitated diffusion, which involves a protein. Okay, next thing then, let's talk about active transport. Now, there's two types of active transport. There's primary active transport and secondary active transport. Now, the convention is if you just say active transport, it means primary active transport. Okay, that's whoever started that convention a long time ago, it just stuck. So if anyone ever says to you active transport, they mean primary active transport. If they mean secondary active transport, then they need to say secondary active transport. For our purposes in the course, we really should get in the habit of being very clear and saying primary active transport or secondary active transport. Now, in both cases, you see it's active. What do we mean by active? It means that you got to apply energy into the system because you're moving things against their gradient. Now, you already know this. You should have some intuition about this because let's go back and look at this. Which way do purple dots move? Left to right or right to left? Well, the answer is obvious. If they're left alone, they're going to move left to right, right? We know that because that's how the concentration gradient is set up. The question then becomes, well, is it impossible to move dots from the right to the left? Will that ever happen? And you'd say, well, no, it can happen, but only if I apply energy into the system. If I set up a table fan right over here and I blow it in this direction from right to left, then yeah, those purple dots will move from right to left. But again, I'm applying energy into that system. We've seen this previously in the electron transport chain. Can hydrogen ions move against their gradient? Yeah, of course, as long as you apply energy to that system, which happen to be the energy of electrons moving down their gradient, right? But again, things can move against the gradient, but you gotta have energy. And so active transport means you need to have energy. You gotta have some source of energy if you wanna move things against its gradient, if you wanna move it against its gradient. So now, the difference then between these two lies in the source of energy. In primary active transport, the source of energy is ATP. Boom, there it is, simple and easy. Secondary active transport, though, is a lot more interesting or clever in some ways. So let's look at a picture of those. When you look at something... Let's look at primary active transport first. What we have here is an image. And this image is a little tough to decipher without any setup. So what I've created here is showing you that these little red dots are sodium. Okay, I'm telling you they're sodium, and they are located in high concentration outside the cell. And on the inside of the cell, they're located with a low concentration inside the cell. So which way does sodium want to move? Well, you know which way it wants to move. It wants to move from high concentration to low concentration. So I put this arrow here and said sodium, if it could diffuse, is going to diffuse from outside of the cell to inside of the cell. And for right now, let's just say it's simple diffusion. Okay, we'll look at some other stuff later. But for right now, let's just call it simple diffusion. Now, here's my question. Can I reverse this? Can I reverse this arrow so that, and now you see I flipped that arrow, can I make sodiums move from the inside of the cell to the outside? In other words, can I make it move against its gradient? And you should be nodding your head saying, well, yeah, of course, as long as you have energy, as long as you apply energy into the system. And that's right. So how am I going to do that? I apply energy in the system. In this case, what this image is showing us is that that energy comes from ATP, and that's what this is showing. And it's moving sodium from inside of the cell to the outside of the cell. So here it starts, in the middle, and now at the end. Yay, I move sodium out to the outside of the cell using the energy of ATP. Therefore, this is primary active transport. Okay, so describe the concentration of gradient. We just said that. Which way is sodium moving? Well, in this picture, sodium is moving against its gradient using the energy of ATP. Therefore, how did sodium move? Sodium moved by primary active transport. Okay? So now, secondary active transport. Now, this was the kind of interesting one. I want you to imagine this kind of pulley system. I've made a little hill here. There's this yellow guy and this blue guy here. Now, this yellow guy, if he grabs onto this zip line, then does he have to apply energy into the system for him to go from high ground to low ground, to go from high ground to low ground, that ground gradient, so to speak? And you should say, well, no, all he's got to do is lift up his feet and then gravity takes him down. This is the equivalent of simple diffusion. Now, we wouldn't actually call this diffusion. Physicists would never call this diffusion, but I'm just trying to draw analogies here. This guy moves, no energy required. He just lets go and boom, he goes down the gradient. But what about this blue guy? This blue guy, he's at low ground. Can he go to the high ground? Again, your answer should be only if there's energy applied into the system. Well, if the energy came from ATP, we would call it primary active transport. But where is the energy coming from in this case? It's actually coming from the yellow guy. Because let's call him 300 pounds and he's 100 pounds. When this guy lifts up, then this thing goes right to left, high ground to low ground. And the blue guy on this pulley goes from low ground to high ground. But again, where did it come from? The energy released from the yellow guy going from high ground to low ground. So by definition, what is secondary active transport? It's the movement of stuff against its concentration gradient, using the energy of something else going down its gradient. Okay, so that is secondary active transport. Here's an example of secondary active transport. I've got a high amount of sodium on the outside and actually have a high amount of glucose on the inside. And what I'm highlighting right here is I'm highlighting this thing right over there, because what we're about to see is we're about to see that glucose come to the inside. So how did glucose come down to the sorry, the glucose is the hexagon here. My cursor was on the wrong thing. That glucose is going to come to the inside, but you'll see that, oh, that would mean he's going against its gradient, right? Because a high concentration of glucose on the inside, low concentration of glucose on the outside. So how is he traveling against his gradient? He's doing it by using the energy of sodium when sodium goes down his gradient. Okay, now that's where this analogy falls apart. In this analogy, these guys were moving in opposite directions, but I just created that because I think this pulley system is kind of intuitive to us. But my point is here, the energy comes from sodium going from high to low concentration, and it's able to drag glucose along. All right, so that is secondary active transport. Let's see. Now then, what is our next learning objective? So we now describe the difference between simple and facilitated diffusion. We now describe the difference between primary and secondary active transport. And I strongly encourage you to definitely memorize the definition, but also make it make sense. Lastly, let's define co-transport and the difference between symporters and antiporters. So what's up with that? So co-transport, it's actually pretty boring. It's exactly what what you think it means. It means two things move at the same time. So this are two things moving at the same time. Yep, sodium and potassium. And so that is a symport system as well too. So co-transport is simply when two things move at the same time, like two different things is what I mean. And if things move in the same direction, then we describe that as a symporter. This would be a symporter because sodium is going from outside to inside and glucose is also going from outside to inside. They're both going the same direction, therefore this is a symporter. And what are antiporters? Antiporters are when things move in opposite directions. And so this thing here is an antiporter because we're watching sodium come from outside to inside, whereas calcium goes from inside to outside. And so they're both moving at the same time, but in opposite directions. So this right here is an antiporter. This is another famous example of an antiporter where sodium comes in and hydrogen ion goes out. All right. Now, symporters here, we're seeing sodium comes in and glucose comes in. This image right over there is exactly the same as this image right over here. Okay. And then this where sodium comes in and amino acid also comes in at the same time is another famous example of a symporter. For right now, I don't need you to memorize any famous examples of antiporters and symporters. I just need you to understand that we have co-transporters, and symporters are co-transports where they move in the same direction, two things, and antiporters where they move in the opposite direction. All right. So let's recap where we are. We have covered learning objective number 1. We've covered learning objective number 2. Co-transport is when two different things move across, in our case, a membrane. And the difference between symporters and antiporters is direction, right? Symporters both go the same way. Antiporters, the two things go opposite ways. And that'll wrap it up for this video.